Okay, I think we can start. Hello and welcome everyone for today's webinar on the evaluation of chemical-induced cardiotoxicity, the challenges and progress. This webinar is organized by Work Package 2 partners of the European Horizon 2020 project Alternative, which aims to develop non-animal based methods to detect the cardiotoxicity of chemicals. I am Sivakumar Murugadas, involved in the Alternative project, and I will be moderating this session. We would like to inform you that this webinar will be recorded, and the webinar recording will be made available in our project website. So we highly recommend the participants to turn off your microphone and then video, unless you have something really important to speak. Today, we have three speakers from the Alternative Consortium, and I will introduce them briefly before each presentation. So most importantly, please drop your questions in the chat box. We address one or two questions at the end of each presentation. And also our experts and speakers, they address the questions directly in the chat. And also we will discuss some questions at the end of all presentations and in the Q&A session. And we will also get back to you if some questions left unanswered. Without further ado, I would like to introduce our first speaker, Dr. Nunzia Linzalon. She has been carrying out research activities for more than 20 years in the field of environmental epidemiology at the Institute of Clinical Physiology of the National Council of Research in Pisa, Italy. She has been developing transnational research activities in the field of population health in industrial and contaminated areas and of exposed workers. Today, she is going to talk about the topic, epidemiological evidence for cardiotoxicity induced by chemicals. Nunzia, thank you for joining us, and the floor is yours now.
Hello, good morning. Can you hear me?
Yes, we hear you.
Okay. So thank you, Siva, for your introduction. Going to the basic new knowledge on human cardiotoxicity and pollutant exposures from an epidemiological perspective. So among the research areas carried out in the Institute of Clinical Physiology, the epidemiology provides descriptive and analytical studies on human populations to understand the causal link between disease and the risk factors and their distribution over space and time, adopting the analytical methods of the biostatistics. These skills are supporting the multidisciplinary...
Nunzia, sorry to interrupt.
Yeah?
Please share your screen.
Oh, sorry. Don't know why this is...
Yeah, please try to increase also the volume of your voice, because there are someone that has some problems to hear it.
Yeah?
A little bit low volume.
Okay, and then see if you... Are you looking at my presentation too?
We see your desktop. You can share your screen and then go to your presentation, and then you can turn on the present. Yeah.
So are you?
Yes.
Yes, it's okay
Yes.
now. Yeah,
Yeah.
sorry
Yes.
for the inconvenience. And regarding my volume, I can check my earphone. I hope this can.
Try to stay more close to the microphone.
Yeah, exactly. Yeah, like this. Okay, I can start again. Sorry for the inconvenience. And I will describe the epidemiological perspective uh, and our skills in the Institute of Clinical Physiology are supporting the multidisciplinary team of the Alternative Consortium to develop a more comprehensive framework for the risk assessment of cardiotoxic chemicals. So to start, we know that in the era of sustainability, international policies set among target goals to reduce the number of deaths and illness from hazardous chemicals and air, water and soil pollution and contamination as well. Indeed, currently, almost 70% of deaths from chronic disease are caused by toxic exposures, and cardiovascular disease are leader, followed by respiratory disease, cancer, and diabetes. Scientific evidences are highly supportive of the particulate matter as causal agent of ischemic heart disease and stroke. Particularly, an increment of PM2.5 in short-term exposure has been associated with a 2.1% relative increase in heart failure hospitalization or death. These data are consistent with the 90% of the world's population being exposed to air pollution level exceeding those recommended for healthy stay. When we speak of CVD, generally we use an umbrella term which include a range of pathological conditions affecting the heart muscle and also the blood vessels. However, we need to differentiate the heart disease referring to those conditions affecting the structure of the heart and the way it functions only. CVDs are determined generally by non-modifiable traditional risk factors like age, sex, health, family history, and comorbidities, and also modifiable factors mainly represented by behavioral attitudes. Among modifiable factors, socioeconomic disadvantaged status and environmental pollution exposures are today largely responsible for avoidable premature mortality as a consequence of globalization. But the modern society is also affected by an aged-fashioned population. In particular, Europe has the oldest population in the world. Age, therefore, is a threatening factor not only for the youngest but also for the oldest, more than gender, due to organ vulnerability and to comorbidity. Particularly, an aged heart is sensitive to the exposome detrimental impact, carrying on the lifetime exposure burden to a variety of external and internal stressors from conception onwards. Heart failure is one of the most prevalent cardiovascular disease and a persistent and a strong risk factor for ischemic stroke. So measurable changes of cardiac morphofunctional parameters are prognostically important biomarkers in CVD. A transformation of the left ventricle, making the heart enlarged, dilated, and weakened, inhibits the heart's ability to properly pump the blood, leading to heart failure and ischemic heart disease. Therefore, an early diagnosis of heart failure based on cardiac functional alterations can help reducing the burden on the healthcare system, preventing the disease progression to symptomatic and advanced stages. We know that known and also unknown exposures together start a complex biological cascade of responses after the contact has occurred with an organism. In the alternative project, we focused the pathophysiological mechanism of heart failure. The underlying hypothesis is that the early subclinical signs observed in exposed individuals are attributable to altered myocardial contractility in cell death Unaltered physiology can be referred to a molecular and cellular adaptive response, likely oxidative stress and mitochondrial dysfunction, as a direct effect of exposure. Therefore, we need to improve our knowledge about adverse outcome pathways that are plausibly linked to toxic compounds behaviors, thus helping to explore the mixture's effect and the synergic action occurring under real-world conditions. To date, evidence are strong stating that cardiovascular mortality is associated with air pollution. However, almost not existing knowledge regards the role of low to medium prolonged exposure to chemical mixture on early stages of heart failure. We conducted the first ever systematic review echocardiographic parameters related to left ventricular dysfunction in exposed population to identify subclinical markers of exposure. The protocol for the search was strongly validated following the international procedures and guidelines and was double-checked from peer reviews. It is published, you can find it on the web. The selected studies from the review were consistent with the PICOS defined in the search strategy. We retrieved highly heterogeneous early markers of heart failure, as you can see here in the screen, and we grouped them in the three meaningful outcomes category of structure and geometry, left ventricular systolic function, and the left ventricular diastolic function. Overall, we found 521 estimates of the association between cardiac parameters and environmental pollutants. Of these, 166 showed a significant association with the pollutants categories of air pollution and metals, pesticides, and other persistent organic pollutants. The quality evaluation of the epidemiological studies provided indications for exclusions in the subsequent quantitative meta-analysis. To preserve the statistical significance of the meta-analysis, only the pairs outcome pollutant with a minimum number of 5 comparable effect measures were analyzed. Also, an internal validation of results was performed to exclude inconsistency of results, and furthermore, a sensitivity analysis was done to have a control on the repeated measures we used. The findings confirmed a negligible depressing effect of the particulate 2.5 and 10 on the systolic and diastolic function, and on lead only on the systolic function. These are the results from the meta-analysis, but more details will be soon published. Other significant results were not confirmed after the sensitivity analysis. However, these results suggested for both the particulate matter 2.5 and arsenic in modifying the left ventricular structure, and for the nitrogen dioxide and arsenic affecting diastolic function. To confirm further this suggestion, new adequate epidemiological studies and data are needed. So, limitations have to be considered when interpreting the findings from systematic reviews. We note that academics advise that the systematic review, that is the process of selecting studies, and meta-analysis, that is the statistical aggregation that produces a single effect size, are tools to interpret the evidence. Therefore, we suggest to adopt a hybrid approach to judge the final results, because This approach uses 7 more judgment dimensions to reach a conclusion. Regarding the current knowledge, the very limiting factor of our research to the grading of evidence is the insufficient number of studies currently available, only 15. We also acknowledge that the medium and low level of quality of the studies need improvement, mainly providing data on dose-response relationship. And finally, types of studies apprised being almost cross-sectional should be turned into prevalent longitudinally study to be prognostically significant. Nonetheless, we found signs of increased risks consistently with the literature, highlighting a detrimental effect of air pollution and heavy metals on cardiovascular disease. Even if the findings account of the large number of published historically addressing this kind of pollutants. So, other contaminants are entering the scene and require consideration of their potential cardiotoxicity and synergic effect, which are still unknown but probably likely. It is the case of PCV. So, prospective interpretation of the results suggests that a new epidemiological research issue calls for further investigation to discover how more proximal markers of heart failure are affected by exposure. Therefore, the coming years will be crucial in elucidating the toxicity properties of chemical substances triggering the pathophysiological mechanism responsible of the progression of heart failure. Also, the measurements of echocardiographic alterations are the new frontiers for the observational epidemiology to relate real and lifelong exposures with the occurrence of chronic disease. Moreover, an overarching integrated framework collecting epidemiological and toxicological data is needed to systematically include the contribution of the adverse outcome pathway approach to confirm the plausibility of evidence retreat. Then, going to our conclusions, we suggest that evidence needs to be improved, adopting a new study design, longitudinal and case control, opening to omics epidemiology to investigate the proximal marker of damage, and focusing on the vulnerable category, including fetuses, children, and the elderly. Our advice to regulators, therefore, regard the need to systematically collect evidence on heart, consider the one toxicological target of chemicals, using a new approach methodologies, thus covering all the biological level of information. To decelerate the CVDPs in the aging world, early markers of cardiotoxicity should be monitored in clusters of the vulnerable populations. Actions is also need to minimize the release of those identified cardiovascular stressors in the air and the water mainly. So, I want to thank you for your attention, and I want to thank my collaborators and, from the Institute of Clinical Physiology, Gabriele Donzelli and Morales, but also collaborators for University of Utrecht, Professor Ronette Gehring, and thank you all for your attention.
Thank you, Nunzia, for this wonderful presentation showing us the association between some chemicals, exposure to some chemicals, and heart failure parameters, and also pointing out the research and regulatory needs from epidemiological perspective. So, so far, I want to thank you once again. So far, we have no questions in the chat box, and also we are on time for the second presentation today. I would like to introduce our second speaker, Dr. Alexandra Shafat. She has a background in the toxicological omics-based analysis of endocrine disruptors and their mode of action. She is currently working as a postdoc researcher at the Medical University of Innsbruck, Austria, mainly focusing on building bridges between science and the regulation of chemicals. But today, she is going to talk about the regulatory requirements for cardiotoxicity assessment. Alexandra, thank you for joining with us, and now the floor is yours.
Thank you very much, Siva, for the introduction, and also thank you, Nuncia, for this comprehensive introduction on cardiotoxicity. And yeah, I would like to focus on the regulatory requirements. And we are currently, for cardiotoxicity, in a catch-22 situation. This means that we believe, to our knowledge, that the need for more specific cardiotoxicity assessment has not yet been recognized. And this, in turn, of course, leads to the currently limited regulatory assessment of cardiotoxicity. This then leads to currently the lack of available data on cardiotoxicity, as Nuncia has presented in her talk. And this in turn, of course, causes the need for more specific cardiotoxicity assessment to not be recognized. And to break out of this dilemma is part of the alternative project. And before I come to the more specifics on what the project is trying to do to solve this dilemma, it is necessary to see what do we actually know about the cardiotoxicity of chemicals. And here we have summarized the current textbook knowledge on cardiotoxicity. And it is well known that stressor exposure of the heart can induce and result in myocardial injury. And this can cause maladaptive changes of the heart and the heart tissue. This includes cardiac remodeling. Here we can have either hypertrophy or also fibrosis. Hypertrophy means that the total number of cardiomyocytes reduces while the volume of the individual cells increases. And this causes then the heart walls to thicken. Fibrosis of the myocardium develops through an increase in collagens and various other extracellular matrix components of the heart muscle. And this occurs to an excessive extracellular matrix deposition, as you can see here in blue depicted. And this can be due to tissue injury or cell death. And it is also a common phenomenon in aging. Yeah, upon chronic exposure or very severe cell death, this condition can lead to cardiomyopathy. Cardiomyopathy is a clinical term for diseases of the heart muscle. And this includes when the chambers of the heart have become stretched, thickened, or are stiff, and this then affects the heart's ability to pump blood and can ultimately lead to heart failure. Heart failure is seen as the ultimate cardiotoxic manifestation. And other mechanisms can cause or contribute to heart failure too. This would be also endothelial injury, which can also contribute to myocardial injury. It is also known that stressors can induce arrhythmias, which are disturbances in normal cardiac rhythm. And this then causes QT prolongation, which then can lead to heart failure. There are also indirect mechanisms like hypertension or atherosclerosis that can contribute to heart failure. Altogether, the heart is a critical organ for toxic effects due to the very specific function of cardiomyocytes and their limited proliferative capacity, and yeah, the high remodeling efficiency of the heart, and which is following due to injury. The alternative projects tries to work on assessing cardiotoxicity since October of 21, and the objective here is to develop a novel platform to detect cardiotoxicity of chemicals and their mixtures. The vision is to create a 3D in vitro model that is composed of human-induced pluripotent stem cell-derived cardiomyocytes, and there will be two versions, one mimicking the young and one the aged heart. We plan also on addressing mixture toxicity, and there will be kinetic in vivo, in vitro to in vivo extrapolation for cardiotoxicity risk assessment. This project includes a machine learning in silico cloud system for scientific and efficient data evaluation. And there will also be omics analysis that provides biomarkers for a mechanistic characterization of cardiotoxicity. To achieve all that, we are a very multidisciplinary consortium combining biologists, chemists, bioinformaticians, engineers, omics, and regulatory experts. And the 3D cardiac model that is created using a sensorized bioreactor system, which replicates biological environment. Then there will also be transcriptomics, proteomics, and metabolomics that will help to characterize the effects of single chemicals and mixtures. And through integration with a machine learning approach in silico, we will achieve an evaluation and integration of the biological data. And this project also includes a comprehensive epidemiological and toxicological analysis via systematic reviews of the current state of evidence, as was also described partly by Nuncia in the first talk. And the evidence from that will be integrated into adverse outcome pathways to ensure the regulatory uptake of the knowledge. However, we believe to achieve cardiotoxicity assessment, there needs to be a change of cardiotoxicity assessment in general. Up till now, in general, toxins were assessed by exposure of model organisms, and then adverse outcomes were observed in the whole animal. Those might include mortality, reproduction, or development. However, this approach neglects the mechanisms behind those adverse outcomes and comes with some challenges. Of course, we all know that animal testing is very time and cost intensive, and therefore it is rather unsuited to assess the overwhelming amount of chemicals that currently need to be tested. In addition, problems with the transferability between species and also to humans call into question the relevance of this approach to humans. Therefore, we believe that to solve these problems, there needs to be a change in the toxicity assessment. And this change then can focus more on this black box, the mechanisms behind the adverse outcomes. And here this is explained also general based on the field of developmental neurotoxicity as described by Paparella et al. we normally have an exposure that is first attacking at a molecular level. This can include protein interactions, DNA binding, receptor ligand interactions, and organelle functions. This in turn then triggers a cellular response. This can include transcription, protein production, differentiation, apoptosis, and so on. And this then in turn again triggers a tissue and organ level response of, for example, altered physiology or tissue development and function or damage. This then can lead to adverse outcomes on an organism level that were previously observed in the approach of the past. For example, here again, mortality, reproduction and development. But there are also adverse outcomes that cannot be assessed by animal testing. Also here in general, for example, complex problem solving, sportive or artistic activity, reading and planning, which are very relevant for humans. Additionally, in reality, every response level can be affected by modulating factors. Those modulating factors can be, for example, epigenetics, diet, lifestyle, economic background, stress, and so on. And those factors can either favor an adverse outcome or compensate for it. And assessing those in animal studies is also very hard. Future toxicity assessment can be based on the evaluation of molecular and cellular effects, which represent risk factors for toxicity and disease. And the evaluation can be facilitated using human-relevant new approach methodologies and also NUM-based mode of action hazard classification, and in addition, subcategories for potencies within the class. Within the alternative project, we took the challenge to navigate the way to cardiotoxicity assessment of the future. To achieve this, we first identified limitations and needs in current regulations. We then went out to build a weight of evidence for cardiotoxicity. We are doing this currently by gathering evidence via systematic reviews and integrating and causally connecting those different levels of evidence. And of course, assessing the weight of evidence of chemicals leading to the adverse outcome, which we here focus on the heart failure because it is, according to textbook knowledge, the ultimate outcome of cardiotoxicity. And this is done using the AOP framework, the Adverse Outcome Pathway Framework. This will also include a collection of methods and assays that measure specific key events of the AOPs. It will include computational methods predicting molecular initiating events or key events. And lastly, out of all this, we will draft an integrated approach to testing and assessment. So what are the current regulations that might address cardiotoxicity, and how are where are their knowledge gaps? For methods to specifically evaluate effects on the cardiovascular system are described currently in the ICH guidelines for pharmaceuticals. And they include mostly in vivo testing, which includes evaluation of heart rate, electrocardiogram methods, and blood pressure evaluation and QT prolongation, and in some cases also histopathology. In vitro testing can also be conducted here. The H2020 assay is used, which measures changes in activity of this potassium channel and is able to detect a delayed repolarization, which is a risk factor for arrhythmia. However, the available methods are too limited to sufficiently evaluate pharmaceuticals and, of course, also environmental contaminants. The in vivo guideline studies suffer from a conflict with the 3R goals, are economically and time resource intensive, and are generally subject to uncertainties in extrapolation between different animal species and humans. The existing in vitro guideline studies do not adequately cover all mechanisms like other ion channels, and the assessment of structure and contractility is completely missing here. And in general, there's a very limited predictivity of current methods of in vitro and in vivo testing. For chemicals, pesticides, and biocides, current regulatory guidelines are covered in OECD test guidelines, as shown here, and they include an evaluation of cardiac organ weight, pathology, histopathology, and cardiac malformation. And however, as you see here, those are only in vivo studies, and of course, those suffer again from interspecies difference and extrapolation issues. Moreover, currently high doses are administered in those guideline animal studies in order to increase the chance to achieve significant effect sizes, to derive limit values, and also to achieve a sufficient severity to allow for classification under the current regulations. And as a consequence, multiple effects and also partly unspecific effects are induced, and this makes the identification of specific mode of actions very difficult, to which heart failure might belong to. And also animal tests, of course, are problematic to assess a large number of chemicals that we need to test currently. And this then comes along with high expenses and time, very high amounts of time, and problems with ethics. And therefore, we believe that the needs that arise from those current regulatory gaps are that we need to the development of human relevant non-animal based methods that focus not only on electrophysiological but also on structural and contractility endpoints. And those need to be more cost effective than in vivo testing. They need to cover small chronic doses, complex mixtures, and susceptible populations. We have also identified other regulatory challenges. For example, cardiotoxicity is not addressed as a separate endpoint in the case of chemicals, pesticides, and biocides. And the limited regulatory experience then leads to that the implications on limit values and classification are not well known. Complex in vitro systems, there is also limited regulatory experience with results of those systems, and method validation is not yet included in the regulatory path. Also, those systems are not high throughput. There are also regulatory challenges concerning the elderly population. The proportion of the elderly population is rising. It is predicted to be 22% of the in the whole world in 2050. And we all know that this population has a higher risk for cardiac effects. And however, there is no database assessment factors currently available for the elderly population. Since we are exposed to a mixture of chemicals, this might play also a relevance for cardiotoxicity. However, there are too few mode of action data for individual chemicals available to calculate potential mixture activity, and too large number of chemical mixtures to test via animal tests, which comes along also with high expenses and time consuming, and which is also unethical at this amount. So as the next step, we started to gather evidence for cardiotoxicity of environmental contaminants. We started here with systematic reviews of the evidence based on in vitro and in vivo toxicity data, and also epidemiological data, as Nuncia presented in the first talk. This evidence will be integrated into an AOP framework. And here in vitro mechanistic toxicity data will support the lower part of the AOP. The in vivo toxicity data will also support this part of the AOP up till the organism response level, and epidemiologic data will support organ response level and adverse outcome evidence on an organism and population level. Out of why did we even choose the AOP framework to integrate the data? This is because AOPs support one, the weight of evidence interpretation. They help with assay development that address the molecular initiating event and key events. They thereby help replace animal tests with NUMS, and they also help with the design of integrated iterative testing strategies. And we came up based on the textbook knowledge and the preliminary knowledge from the systematic reviews. We came up with these pathways for the AOP network. Here it includes an electrophysiological contractility pathway, which was created by Luigi Mariotta Casalucci. It is the AOP 261. It can be found in the AOP wiki. And also within the alternative project, we created the structural and contractility pathway, which can also be found on the AOP wiki. And all those pathways come together through a decrease in contractility, which leads to heart failure. And the vision is that through the 3D model that is developed within the alternative project, we can have here a functional anchoring. This contractility can be assessed in this 3D cardiac model, and the project will also provide biomarkers of effect to assess those key events at the beginning of the AOP. And from this, we are planning to draft an integrated approach to testing and assessment. What is an IATA actually? What is the intention of an IATA? It is to internationally harmonize assessment procedures and thereby also optimize their speed and efficiency. And mechanistic knowledge summarized in AOPs can explain the mechanistic relevance of an IATA and be the basis for testing, as shown here with assays measuring the MIE or the specific key events. And upstream molecular and cellular effects can then indicate a likelihood for downstream adverse outcomes. So our vision of an IATA that is also covering cardiotoxicity assessment might look as follows. First, we could start with an assessment of all relevant available information. Of course, those sources need to be characterized in guidance, and they might include in silico predictions, literature, so systematic reviews, QSARs, ADME, and PBK modeling. And for the first step, a weight of evidence assessment will be conducted, and if possible, a regulatory decision could be derived here already with classification and limit value. If a conclusion cannot be made in the first step, further data might be necessary to assess the risk. And this then can include further assays starting from rather untargeted, simple and fast assays and methods, and further going to more complex and detailed assays. This can include, for example, at the beginning, an untargeted screening to do effect identification and hypothesis formulation. And for example, this can include broad testing with a threshold for toxicological concern, TTC approach, with high throughput screening, with in silico predictions for AOP, MIEs for with omics and bioactivity NUMs. And on this basis, a mode of action hypothesis could be built to be tested with more complex 3D or 2D models or organ on chips. And this then could include, for example, a testing for cardiotoxicity. And experimental data also needs kinetic, quantitative in vitro to in vivo extrapolation, and the mechanistic knowledge used for very systematic testing here could be derived and based on adverse outcome pathways, and here MIEs and key events of the adverse outcome pathways could be tested. And this would then again go to a weight of evidence assessment and be stopped when a regulatory decision can be reached. And here the NUMs could also provide then comparison or information on young versus aged heart. And lastly, the IATA needs to be validated. The validation of an IATA means the characterization of robustness of individual assessment methods and mechanistic knowledge. And the alternative project is contributing to several elements of this IATA, besides, of course, drafting it. And with that, I would like to thank you for your attention, and I would urge you to please scan this QR code. It has some questions to you, and those answers, because your feedback would really help us to tailor our approach and maybe thereby change the future of regulatory cardiotoxicity assessment. We have just a few minutes left till the next talk. Maybe you would like to grab a really quick coffee and start answering those questions. That would be absolutely amazing. I will also post a link in the chat so that you can click on there. Thank you very much.
Thank you very much, Alexandra, for the nice overview of existing regulatory requirements for cardiotoxicity and the knowledge gaps and how we address them in the alternative project systematically. So now, as you said already, meanwhile, while people are answering this poll. Yeah, we have a coffee break for about, let's say, until 5 minutes before 1, or just we come back at 1. Alexandra?
Yeah.
So we will come back at like 5 minutes before 1, or just we come at 1.
Let's do 5 minutes. 5 minutes before 1.
Okay,
5.
so we will come back 5 minutes before 1 p.m.
And your questions will be answered at either in the chat or at the end of the webinar in the discussion. Thank you.